Hi.
Hello, are there any questions?
One quick question.
Okay,
What would for the extra credit assignment, do you want all of the group to have one document and each person in the group turn in that same exact document, or only one person in the group and one document for the entire group and the other person not turn in anything?
you only can turn in the document with everybody's names in the group. But if more people turn in the document, that's okay too. Is that clear?
Yes, thank you.
All right, yeah. Since your name will be there from other people in your group, I only need one person in the group to turn it in. So to make sure you get credit, make sure one person turns it in. But the others may or may not. All right, can you guys hear me with the headphone on now?
Yes.
Okay, good.
Professor, will the answers to the quiz be up in a day or?
Can you repeat that? For some reason, I didn't hear that.
The answers to the quiz, will they be available in a day?
They're usually available 2 days after you take the quiz. So this is 1 day
Okay.
afterwards. So they should be available, I think, at 8 p.m. tomorrow.
Okay.
Okay. All right, today is April 21st. Oh, I'm not showing this, am I? Let me share the screen. I am recording. There. Today is April 21st, and we are supposed to be covering chapter 3 and finishing chapter 3 today. We'll see if we manage that. Let me tell you that there is no lab for today. I will be logging in at, is it 6:30? Is that when our class begins, the lab? Let me scroll up here. Yeah, I'll be logging in at 6:30 and I'll be there until 6:45 to answer questions. But if there's no questions or I've already answered all the questions at 6:45, I'll be logging off. The point is I will be there for 15 minutes to answer questions. So make sure if you have questions that you log in between 6:30 and 6:45. But there is no lab, so you do not have to log in. But if you have a question, log in during that time. Any questions about any of that?
So there's no lab due this week?
Say again? I'm not
Is there
hearing too well.
no-
I don't know why. Can you
Is there
repeat that?
no lab due this week?
We did lab 4 on Tuesday. So that
Okay.
has to be turned in on Saturday, 11:59 p.m. Saturday night.
Okay.
Okay. All right, so chapter 3. Let me get to where we left off. So we had talked about the typical size of microorganisms. When we're talking about the size of microorganisms, you should realize that they fall on a spectrum of what you can see with your unaided eye. Let's scroll up here. Oops, too far. So obviously you can see a person with the unaided eye, a chicken egg. You can also see frog eggs with the unaided eye. And you can see a little smaller than that. And this is the region that we use a light microscope. And it can see things from about a frog's egg down to a eukaryotic cell, which is about 10 times bigger than your average bacteria cell, which is about 10 times bigger than your average virus. Below that, a virus would be a very, I don't know, it's not even a molecule because it's many molecules. A large object compared to a large molecule. So underneath that would be a large molecule, like a protein. Underneath that would be a small molecule like water. And then underneath that would be atoms. And this has the size of the things on the left. You don't need to know the size of the things on the left, but you should know the relative rank, meaning what's larger than what. And note that this graph, which I don't know why Pearson never corrected this, it's got an error in the graph. It's got 1 micrometer right there, and that should actually be 1 millimeter. That's my correction.
I have a question, Dr. Volpe.
Uh-huh.
Or on the quiz, the lockdown, the multiple choice, there was a question where we had to arrange things from smallest to largest.
Uh-huh.
And now looking at this diagram, I feel like this was probably from chapter 3.
This picture, this graph was both in chapter 2 and on chapter 3. You might not remember it from chapter 2, but it is in chapter 2 right here, the scale of things.
Oh, okay.
And so that's why the question was on the quiz.
Okay, thank you.
Okay. We do repeat a number of topics in this class. You will be covering the Cox postulates 3 different times in the class. We will be talking about endospores. I think we'll be talking about them 3 different times in the class. And the cell wall of bacteria, I think we talk about them 3 different times in the class. And so we do cover a lot of important topics more than once. And you may get the quiz question earlier or later. The point is, it's an important topic, and we will cover these topics again and again, or at least some of them. On chapter 3, we're going to talk about the compound microscope. You've already talked about it in the lab. A compound microscope is one that has 2 or more lenses that magnify. So the compound microscope has an ocular lens, or the eyepiece, which magnifies, and an objective lens down here, which also magnifies. All of our ocular lenses magnify 10 times, but you can find microscopes that can magnify at different magnifications. Most of them can magnify up to 20 times if they're different than 10 times. The ocular lenses can vary depending on which objective you're using. Not the ocular, the objective lens can vary depending on which objective you're using. On our microscopes, we have a 4x objective, a 10x objective, a 40x objective, and a 100x objective. The total magnification is the objective magnification times the ocular magnification. You should know the different parts of the microscope, and some of them are labeled here. Shrink that a little bit. This is a little better. You should know the ocular lens, also called the eyepiece, the tube or the arm right there. The objective lenses, the stage for holding the slide, the stage clip for holding the slide on the stage, the condenser, the iris diaphragm. And you'll notice that this microscope is different than the one picture I showed you in the lab. They're still together, but in the last one, the diaphragm was above the condenser. Here we have the diaphragm below the condenser. The illuminator, also called the lamp or the light source. And on some of your quiz questions, people said that the illuminator was the important part of the microscope for adjusting the intensity of the light. You can't adjust the lamp. You can adjust the illuminator by either using the iris diaphragm or by using the rheostat, which is called the light, also called the light intensity knob. Okay, the base, which you hold the microscope by the base and the arm. The coarse focus knob is the big one, the fine focus knob, the small one. Now I've showed you this once before. I'm showing it to you here to emphasize a simple microscope. This is actually a microscope that was built by Anton van Leeuwenhoek. And it's a simple microscope because it only has one lens to magnify. So in a sense, this is really a super duper magnifying glass. That's what we're going to call it. But it's also called a simple microscope, and a simple microscope just has one lens for magnifying. The resolution of a microscope is the ability of the lenses to distinguish 2 points as separate points. So like if they're touching each other, you can't say they're 2 points, but if you can distinguish them as 2 points, that is the resolution. A microscope with resolving power of 0.2 micrometers can distinguish between 2 points that are separated by at least 0.2 micrometers apart. And this is actually the resolution limit of white light, approximately 0.2 micrometers. You cannot magnify above this because of the properties of light. So the resolution limit of all light microscopes is 0.2 micrometers. Because even if you could magnify further, the properties of light do not allow you to resolve 2 points that are separated by less than 0.2 micrometers apart. The best compound light microscope has a total magnification of 2,000 times. And I'll warn you, in Clark College, in the microbiology lab, we do not have the best microscopes. Okay? Any question about any of that?
I have a question about the, you said the illuminator. What was the answer about how we changed that? Was it the fine focusing knob?
That's one of the questions in the lab, so I don't really want to say how you do it, but the lab manual does discuss it, and in fact it has it and says this is the best way to adjust the light. Okay.
Okay.
All right, so the lab manual does discuss that. So these are 2 points right here that we're looking at under the microscope, and they're separated by 200 nanometers, or the limit that light can distinguish 2 points. And we can observe that they are blurry, but 2 separate points. These points here run all together, and we cannot distinguish them. And that's because like this point and that point are separated by less than 200 nanometers, or what is it, 0.2 micrometers. Any question about any of that? All right. So when we're talking about microscopes, another topic we have to talk about is refraction. Refraction is the bending of light. The refractive index is the light bending ability of a medium. So when we're looking at the pencil in water and air, we'll note that the pencil gets bent right here. And that's because light has a different refractive index in water than it does in air. And the pencil is actually straight, but when we look at it under this picture, it looks bent because of the difference in the refractive index between air and water. And whenever water changes from one medium to another, the light can bend if the two different media have a different refractive index. So water and air obviously have a different refractive index. We're not seeing it, but glass and water and air all have a different refractive index. Refraction is extremely important for life. We're not going to talk about this much, but like a bird trying to catch a fish actually sees the fish in this location, even though the fish is actually down here. And birds that are fish catchers instinctively know that where they see the fish is not where the fish actually is. And so it will compensate for the refraction of light to catch the fish. The refraction of light is important in the microscope. And as I mentioned, whenever light passes through a media, light bends. So when light comes through glass and then hits air, some of the light will bend. And some of the light will bend at such an angle that it will not be captured by the objective lens. And that's the advantage of using oil. And that is oil, the yellow location here, has a refractive index the same as glass. And so light coming through the slide, coming through oil, will not bend. And then this ray and this ray will be captured by the objective lens. Whereas if we were not to use oil, this ray here would be bent at the interface between the glass and the air, and also between the air and the objective lens. And then this ray here, this one here, would be bent at the slide and the air, and would be bent away from the objective lens. And the reason why oil is used on a light microscope, particularly with the oil immersion lens, is it allows the light doesn't refract. And so more of the light comes through the slide, through the specimen, and can be collected by the objective lens than if you did not use oil. Less light would be collected by the objective lens. Because more light is collected, we have a better resolution of the image. And therefore you can magnify at a higher numerical aperture, meaning you can your magnification can be greater if you're using oil than if you're not using oil. Any question about any of that? All right, bright field illumination is what all of the microscopes in the microbiology lab at Clark use. This is just a regular light microscope. In bright field illumination, the object is lit up by the specimen, and the background is also lit up, why it's called bright field. And what's happening is the light is passing through the specimen as well as through the slide, and then both of the light get to the objective lens. And so we see the object. It is lit up, but it's purple, a little bit darker because it's dyed, a little bit darker than the slide, which is also lit up. Any question about that? Bright field illumination contrasts with dark field illumination. Dark field illumination has a light source, and in between the light source and the sample, we have an opaque disc. Let me blow that up a little, which blocks the light from directly coming from the lamp and coming to the specimen, and then going to the objective lens. And that's why the background looks dark. All of the light coming from the lamp to the specimen or the background of the specimen is blocked by the opaque disc, why the background looks dark. However, some light comes from the lamp at a discrete angle, different angle, and then hits the specimen and then is refracted. And so that refracted light comes to the objective lens. And so only the light which comes through the specimen, which is then refracted, is collected by the microscope. And that's why we see only the specimen. And it's a little bit darker than here because there's less light being collected by the objective lens. And there's no light being collected from the background of the slide. That is dark field illumination. Any question about any of that? Both of them allow you to see the object, but if this was not stained, it would have a similar appearance as the background. It'd be very hard to see the difference between this and that. Dark field illumination is a little higher contrast because the background is totally dark. All right, any questions about the light microscope? If not, let's move on to the electron microscope. We're seeing here, obviously it's much bigger than a light microscope. It uses a beam of electrons to view the specimen. And it's focused, the beam is focused by electromagnetic lenses. They're not glass lenses. Electrons have a shorter wavelength than light, 100,000 times smaller. And so it allows greater magnification, giving greater resolution. And it allows you to see smaller objects like viruses, the internal cell structures, that you cannot see with a light microscope. There are two main types of electron microscopes. There are at least four types of electron microscopes, but there are two main ones. Transmission electron microscope, or TEM, blow that up, which we're seeing right here. And you'll notice that transmission electron microscope shows an object in only 2 dimensions. And when we're looking at a specimen, we're looking at only one plane of the specimen, meaning we're seeing only 2 dimensions of the cell. The advantage of transmission electron microscope is you can see inside a cell, but you cannot see obviously all regions of the cell because you're only seeing one plane of the cell. The second main type of electron microscope is scanning electron microscope. And this is an object here. And you'll notice that it's very different. Number one, we're seeing the object in 3 dimensions. with scanning electron microscope. The other thing is with scanning electron microscope, you can't see inside the cell where you can in transmission electron microscope. With scanning electron microscope, you only see the surface of the cell, but you see it in 3 dimensions. Any question about the electron microscope? Another picture of electron microscope. Scanning electron microscope, we can see this crystal in 3 dimensions. This is a tranning electron microscope. This is a membrane. I think it's the chloroplast of the cell. As you see, it's 2 dimensions. There's also scanning tunneling microscope. We're not going to discuss it very much. You're not going to be examined on it. It uses a metal probe to scan a specimen, but it can get down to 1/100th the size of an atom. So this can magnify very, very small. And then the 4th type, atomic force microscopy. We're also not going to talk much about it. You're not going to be examined on it. It uses a metal and diamond probe inserted into the specimen and allows you to see images in 3 dimensions. And this is actually the toxin for Clostridium perfringens. All right, any questions about the electron microscope? And remember, it allows you to see things much smaller than a light microscope can see. All right, here we have cells on the left and similar cells on the right. Which of these cells are easier to view?
The right side.
The
the
right,
right
the
side.
dark field one.
No, this isn't dark field. That's a good guess.
Oh,
But you see how it's being lit up on the background and lit up on the background? Both of these are using a regular bright field compound light microscope. The only difference between these cells and these cells is that these cells are stained. The dye has been applied. These cells are not stained. And we can see the cells. Like here's a cell right here, and there's a cell there, but it doesn't show up very well from the background. And these cells show up much better. The only thing a stain does is it increases the contrast between the cell and the background of the slide. That's all a stain does, but that allows us to see the cell much better than without the stain. Any question there?
I have a question, not necessarily on specifically what we're talking about, but the
Uh-huh.
difference between the one that's stained and not stained, are those other ones like ghost cells or like possibly another type of bacteria that doesn't work with the same stain? Or
You're talking about like this cell right here?
no, like if you look at the one on the right, how some of them are,
Oh, yeah,
and
these
then
over
some of
here
them
and
aren't?
those right there.
Yeah.
No, those are just cells that are they're out of focus.
Oh, okay. Thank you.
Okay, that's why that cell is so sharp. That one's a little bit blurry. That one's very blurry. And these you can barely see because they're way out of focus. Okay, so this is probably a wet mount. And so the cells are at different locations in the wet mount. But good question. There we go. Most microbes are colorless and have little contrast with the slide or with their medium. Therefore, to see them better, we do need a stain. Any question about that? At least with a regular microscope, I'll word it that way. You could use a phase contrast microscope And we're not going to talk about that in this class. You can read about it if you're interested, but I won't be quizzing you on a phase contrast microscope. All right, so mainly to see an object, we stain them. Staining is using something that colors the specimen or the background to increase its visibility by increasing its contrast. So all the stain does is increase the contrast between the background, which is usually the slide, and the specimen. A smear, you'll recall from the lab, is just a thin film of a solution of microbes on a slide. So when you're making a bacterial smear, you're adding bacteria to the slide. And that's all you're doing. You're not applying the stains yet. Anyways, you make a smear prior to staining the microbes. You also usually air dry the slide and then heat fix it. And that's to attach the cells to the slide. And then an advantage is that that kills many of the cells. Doesn't kill endospores, but all of the vegetative cells would not survive the air drying and the heat fixing. Now, is that true? I should say most species, because there are a few species that will survive the heat fixing, like Mycobacterium. Any question about that? Usually you fix the cells to a slide by adding heat. Stains, also called dyes, are salts. We talked about what a salt is, something that has a positive ion and a negative ion. And there will be a positive, well, I already mentioned that. In the dye, one of the ions is colored. This we call the chromophore. A basic dye has the colored portion in the positive cation, or the positive part of the dye. So the chromophore is a cation. An acidic dye has the chromophore in the anion, or the negative part of the dye. Any question about that? Remember, I've already talked about that. When I have this here? I just think I didn't go down the slide. Oh, maybe I got to read here. Most dyes that we use in microbiology are basic dyes. And that's because bacteria have a slightly negative charge. And so the basic dye is positive, and then it'll be attracted to that negatively charged cell. And so the cell will pick up the dye and be colored. The background of the cell will either be uncolored or will be very lightly colored. So basic dyes tend to be better for staining the cell just because they're attracted to a negatively charged cell. Some examples of basic dyes include crystal violet, methylene blue, and safranin. And we've talked about all of these in the lab. An acidic dye is one that has the negative part being colored. So the chromophore is the anion, and it typically stains the background of the cell, meaning the slide. It does not stain the microbe. Some examples of an acidic dye are eosin, acid fuchsin, and nigrosin. When we stain with an acid dye, it's frequently called a negative stain because we're staining the background instead of the cell. And this term comes from old film. I think they were old Polaroids, but maybe even before Polaroids. They used to make a negative and it had the opposite color of the photograph. Any question about any of that? Both a negative stain and a basic stain show up the cell. So a basic stain stains the cells and either stains the background very lightly or not at all. A negative stain stains the background of the cell, but not of the cell. And as you can see, both show the cells very well. Both of these are a simple stain. Let me remind you that a simple stain is when you're using only one stain to stain the cells. There are three main types of stains: a simple stain, a differential stain, and special stains. So a simple stain is one that simply uses a single dye, and I should actually remove that. A negative stain is a simple stain. That was just a mistake in the slide. Not mine. I got this from the publisher, actually. The primary purpose is simply to increase the contrast between the background and the specimen. The stain is applied to a fixed smear for a certain length of time, and it's usually washed off, dried, and then examined. A simple stain or even a differential stain can use a mordant. A mordant is used to intensify the stain. For example, we talked about iodine in the crystal violet stain being used as a mordant. When we use crystal violet, it increases the affinity between the stain and the specimen. The mordant is not a stain. Crystal violet and iodine complex together and make a bigger molecule, making it easier to see the specimen. With the flagella stain, we can coat the specimen many times so that you coated it enough times, so therefore you can then see the flagella. And that coating is also a mordant. Come on. Recall that a differential stain is a stain that reacts differently with different types of microbes. So that when you're looking at two different cells under the microscope, if you use a differential stain, you can distinguish between the two cells. Usually, two or more dyes are used in a differential stain, but this is not required. The most commonly used differential stains in microbiology are the gram stain and the acid-fast stain, both of which use two different stains. All right, let's talk a little bit more about the gram stain. It was invented in 1884 by Hans Christian Gram. You don't need to know the date, but you should know that the gram stain was invented by Dr. Gram, and that's why whenever we use the word, we use a capital G to describe the gram stain. It's a very useful stain in bacteriology because it essentially classifies bacteria into two very large groups: gram-positive cells, shown here, and they stain purple, and the gram-negative cells shown here, and they stain pink. The difference between the gram staining has to do with the difference in the cell wall, and we'll talk a little bit more about that in this lesson and in the next lesson. How you do the gram stain is you always first make a bacterial smear on the glass slide. Generally, we always do that before we stain the cells. And then you air dry and heat fix. That's simply to attach the cells to the slide. You then flood the slide with crystal violet for 1 minute. If you didn't do the air drying and the heat fixing, when you add the crystal violet, you'll lift up the cells. And then you dump that off and then wash with cold water. And if you didn't do the air drying and heat fixing, that would remove almost all of the cells from the slide. So that's the purpose of the air drying and heat fixing. After you wash with cold water in the gram stain, you then flood the slide with iodine, which is a mordant, once again for 1 minute. Then you dump that off and then wash with cold water. Now you flood with the acetone ethanol agent, which is a decolorizing agent, and you only do that for about 5 seconds. I don't know if you can see this. If you can, what the best thing to do is to view the slide when you're adding the decolorizing agent. And when the color stops running, when you're adding the decolorizing agent, stop adding the decolorizing agent. Any question about any of that? This is the most tricky step of the gram stain. You then need to wash immediately to remove the decolorizing agent from the cells. If you do not wash immediately, the decolorizing agent will continue working and it may remove the crystal violet iodine complex from gram-positive cells. So you do need to wash immediately. You then flood the slide with safranin, and this is the second stain we're using, so we call it a counter stain. And it's applied from 1 to 2 minutes, or 1 or 2 minutes. Okay. You then dump the safranin off and then wash with cold water, blot dry, and then you can examine your cells under the microscope. Looking at the gram stain in a little more detail. When we first apply the crystal violet, all of the cells, gram positive and gram negative, pick up the crystal violet and stain bluish purple. We then apply the grams iodine and the cells will stain a little darker purple, a little less blue. They'll have a little more stain to them. So it'll show up a little darker, and that's not shown here. But they are staining a little more intensely. And that's because the iodine complexes with the crystal violet and makes a larger molecule in the cell. We then apply the decolorizing stagent, the alcohol acetone wash, or it could be just alcohol, or it could be acid alcohol wash. And that pulls the crystal violet iodine out of the gram-negative cells, but does not pull it out of the gram-positive cells, at least if this step is done correctly. And because we can now see a difference between the gram-negative and the gram-positive cells, we say this is the active step or the important step. There's another word I wanted to use here, and I'm drawing a blank at what it's called. This is the differential step in the differential stain of the Gram stain. At this stage, we can see differences between gram-negative and gram-positive cells. We then apply safranin, and then that will stain the gram-negative cells pink, and therefore we can see them better. Okay, any questions here?
On the steps for the washing on the previous screen, was that all washed with cold water when it just said wash?
Yeah, when you wash, except with the decolorizing step, is calling it alcohol wash, that means decolorize. And after each step, right here, right here, right here, and right here, you have to do a water wash. You
Okay.
rinse, in this step, the dyes off of the slide, and here to rinse the iodine off, here to rinse the decolorizing agent off.
Okay.
And when you give the procedure, you don't need to say all of this. You can just say flood with crystal violet, and then after each step, do a water wash. And you don't have to do the water wash after each step, as long as you state someplace, either in the start or the end, after each step, apply a water wash. All right, so when we apply the crystal violet, it does stick to the peptidoglycan in the bacteria cells. Peptidoglycan is only found in the cell wall, so it starts staining the cell wall. Crystal violet is considered the primary stain since it's the first stain we use, and it colors all of the cells, purple or blue. Any question about that? So both the gram-positive cells and the gram-negative cells, after the application of crystal violet, stain purple. We then apply mordant, also for 1 minute. It increases the affinity of the crystal violet for the specimen. The color of the cells after the mordant it's a little more purple than blue purple, both the gram-positive and the gram-negative cells. So it's a little more purple than with the crystal violet alone. But other than that, it's very similar in color. We then use the decolorizing agent, and that can be acid ethanol mixture. It can be an acid ethanol mixture. It can be also just pure ethanol, depending on what you're doing. If you use the acetone ethanol, which is what we use in the microbiology lab, you want to decolorize for only about 5 seconds. It's important not to overwash, because if you overwash, you will pull out the crystal violet iodine from the gram-positive cells, or the cells that you shouldn't pull it out of, as well as the gram-negative, which you should pull it out. It's also important not to under-decolorize, because if you under-decolorize, you will not pull out all of the crystal violet iodine complex from the gram-negative cells, which are cells where the dye should be pulled out. This step is considered the differential step in the Gram stain, because you can see differences between the cells. So gram-positive cells, after the decolorizing agent, stain purple. The gram-negative cells are colorless. We then apply safranin. Safranin is a pink dye. It does stain all of the bacteria cells. It's also called the counter stain because it has a different color than the primary stain, which was the crystal violet. You do apply all of the stains in my preparation for 1 minute. Okay. And it's only the colorizing step, which is less, which is only about 5 seconds. And then you water wash. At this stage, we see that the gram-positive cells retain the purple color, but the gram-negative cells have picked up the pink color. Here's a question for you. I just told you that the safranin stains all of the bacteria cells. Why are the gram-positive cells not staining pink if safranin is in the gram-positive cells?
Is it because purple's darker than the pink?
Yeah, that's exactly it. To our human eye, it looks purple to us because the purple shows up better. And so the human eye only sees purple. But if we were to look at the cell under an electron microscope, we would see regions which are purple and regions that are pink. Okay, but to see the safranin in the gram-positive cells, you have to use an electron microscope. And then you could see the safranin in the cells. All right, any question about the gram stain? You then blot dry the slide with bibulous paper, and then you can examine it under the microscope, including with 100x objective using oil. So the gram-positive cells are purple after the gram stain, the gram-negative cells are pink. And the why? What is the basis for the differential staining? It's because the gram stain stains the cell walls differently. And the gram-positive cell wall is different than the gram-negative cell wall. Essentially, the gram-positive bacteria have a thick peptidoglycan layer. We'll talk about what peptidoglycan is, but it's a disaccharide and amino acids. And the crystal violet iodine complex cannot get out of the thick peptidoglycan, or cannot get out quick enough. And so when we use the gram procedure, it remains in the cell wall. Now, as I mentioned, if you use the decolorizing agent for longer than you should, you will start to pull the crystal violet iodine out of the gram-positive cells. So that's why the decolorizing agent has to be applied for a certain length of time. And if you apply it too long, your gram-positive cells will not stain purple because the crystal violet iodine will be pulled out of them. The gram-negative bacteria, on the other hand, have a thin peptidoglycan layer in their cell wall, and that thin peptidoglycan layer is not enough for the cells to retain the crystal violet iodine. So it washes out of the cells. The gram-negative cells also contain a lipopolysaccharide layer. It's part of the cell wall. We'll talk about that in just a little bit. These next slides coming up. And what happens in the lipopolysaccharide layer is actually the alcohol acetone wash will destroy that lipopolysaccharide layer, meaning lipids are dissolved by alcohol and also by acetone. And so this lipopolysaccharide layer, which is part of the cell wall, will be destroyed in the gram-negative cells. All right, so let's talk a little bit more about the gram-positive cells and the gram-negative cells. Here we're looking at the cell wall. Let me blow this up. And you can see the cell wall, the peptidoglycan layer is thick purple layer for the gram-positive cells. And there is a peptidoglycan layer in the gram-negative cells, but it's much thinner than in the gram-positive cells. And then the yellow underneath is the cell membrane. We've already talked about that. When we look at the gram-positive cells, let's see if I can blow this up. You'll note that it is made up of peptidoglycan. That's the main molecule in the cell wall, both of the gram-positive and the gram-negative. But much more so, very much the main molecule in the gram-positive cells. It's many layers. This picture is only showing 3 layers of peptidoglycan. Come on, mouse. It's actually many more layers than 3. It's like on the neighborhood of hundreds, maybe even thousands of layers of peptidoglycan. It does have other molecules in it, like teichoic acid and lipoteichoic acid. The main reason for these other molecules is to help stick the peptidoglycan together. So the teichoic acid runs through the peptidoglycan and then helps anchor it all together. Lipoteichoic acid does the same thing. The teichoic acid portion of lipoteichoic acid runs through the cell wall, through the peptidoglycan of the cell wall. And then the lipid portion of lipoteichoic acid binds to the cell membrane. So the lipids are inside the cell membrane, which is mainly, well, the inside is fatty acids, which loves lipids. Was somebody going to ask a question? I heard some noise there. All right, the gram-negative cell shown here has a thin peptidoglycan layer and the cell membrane shown in yellow here. And out here, that's the outer membrane of the cell wall, which is lipids, mainly lipids. I should say mainly lipids. When we look at the cell wall in detail, this picture only shows 1 layer of peptidoglycan. That is not correct. There are several layers of peptidoglycan. It's many less than a gram-positive cell, but it's more than 1. Everybody got that? And I'm just going to say several. I'm not sure what the upper limit is, if it's 30 or 100 layers, but it's more than 1 layer of peptidoglycan. Come on, mouse. For some reason, my mouse died. There it is. You'll note we do not have teichoic acid in the gram-negative cells, but we do have a bunch of lipids and I guess carbohydrates and proteins in the cell wall. Let me blow that up a little bit more. This is the cell wall, and it includes the peptidoglycan layer and the outer membrane of the cell wall. The outer membrane is called that because it looks like a membrane, and essentially it acts like a membrane too. The main molecule of both the cell membrane, come on mouse, and the outer membrane of the cell wall is phospholipid. And that's what we're seeing right here. Let's go way to the bottom here. Right here is a phospholipid molecule with, with the phosphate head facing inside the cell, and then another layer of phospholipid outside of that with the phosphate head facing outside the cell. And then the fatty acid being inside the facing inside the cell membrane. We do have the phospholipids in the outer membrane, and that's clearly the main molecule in the lower portion of the outer membrane. We do have other molecules like proteins, especially the porin protein, runs through the outer membrane. On the top portion of the outer membrane of the cell wall, the main molecule is phospholipid, and there we have one here, but we have other molecules in the top portion of the outer membrane. We have this big blue blob, the mouse died again, right here and shown right here, and that is lipid A, and it's hooked to an old polysaccharide, the little hair attached to the lipid A. And so together, the lipid A and the old polysaccharide make a lipopolysaccharide. Lipid A is the important part of the entire outer membrane. Come on, outer membrane of the cell wall. And that's because lipid A is an endotoxin. And as you probably know, a toxin is something that is toxic to our cells. And so lipid A is toxic to our cells. Oh, come on, mouse. My mouse died again. All right, any question about any of that? Let me end the show here. So the crystal violet iodine complex forms in all cells, the gram-positive and gram-negative. The crystal violet iodine complex is larger than crystal violet or iodine alone. During the alcohol acetone wash, the alcohol acetone washes the crystal violet iodine complex out of the gram-negative cells because the peptidoglycan layer is thin, and then the lipopolysaccharide layer is disrupted by the alcohol wash. And so the cells appear colorless because they have nothing to stain them after the alcohol acetone wash of gram-negative cells. In the gram-positive cells, the crystal violet iodine cannot be washed out because it's a thick peptidoglycan layer, and the cells therefore retain their purple color. When we look at the gram stain, we will see something like this, where you see gram-negative cells staining purple, and then gram-positive cells staining, did I say that right? Gram-negative cells staining pink, and gram-positive cells staining purple. There we have a gram-positive rod, a gram-positive cockeye. Here we have a gram-negative rod. Anyone see a gram-negative cockeye? I don't see one here, but there's a gram-negative vibrio, and there's a gram-negative spirochete or spiral. Yeah, I guess that's a spiral. It only has what, 1, 2, 3 bends in it. All right, any question about any of this? When you're looking at the gram stain, there will be a lot of hair pulling, meaning a lot of complications, I guess. I don't know how to word that. Here's one here. And you'll notice that, how do we read this? These cells sort of look pinkish, but these cells right here, next to this black blob or purple blob, look purplish. And how do you read that? When you have a slide you ignore the bad regions of the slide and then read your slide where you have the good regions, which, let me shrink that down. Good regions, easy to distinguish individual cells, easy to see the color. And you'll note that right here, what that is actually a crystal of crystal violet, meaning the crystal violet precipitated at a solution, formed a crystal, and then that crystal got put on the cells. And that crystal has crystal violet come off of the crystal and then can stain the nearby cells purplish. So don't read these cells here because they're only purple because they're getting crystal violet from this solid precipitated crystal of crystal violet. Any question about any of that? So read the slide in the regions where it's good. And then you'll learn that gram staining, of course, you're not going to do it in reality, but if you were, you'd learn that it's kind of difficult. The first time you do it, even the couple of first times you do it, and it's like pulling your hair out because it's so difficult. How you get good results with the gram stain is it has to be performed on young growing bacteria. Any bacteria that are over 2 days old, you should not use the gram stain on them because they may not stain correctly because the cells are too old. Some bacterial cells stain poorly or not at all. That would be the mycobacterium. What happens is they have mycolic acid in their cell wall, and mycolic acid doesn't allow water or water soluble molecules into the cell. And so the cells actually do stain, but they stain poorly. They don't stain as well as other cells. And when you do the unknown project, that will be apparent to you that the gram stain is not as reliable as we would like it to be. The structure of gram-positive cells and gram-negative cells not only affects their staining, it also relates to how they respond to different antibiotics and other properties as well. The gram-positive bacteria, for example, tend to be killed easily by penicillin G. And that's true for gram-positives are killed by cephalosporin and many detergents. Whereas the gram-negative cells are resistant to these particular antimicrobials like penicillin G and cephalosporin. What's happening is, let me come up here. Right here. Is that where I want to go to? Yes. Penicillin G is a water-soluble molecule, and it can move right through the cell wall. Anything that's water-soluble can move through the cell wall. Blow this up. Penicillin G, when it comes to a gram-negative cell, it cannot move through the outer membrane of the cell wall. So the outer membrane of a gram-negative cell protects the cell from the antibiotic penicillin G. And we'll talk a little bit later about what penicillin is doing. It's actually interfering and damaging the peptidoglycan in the cell wall. And if the penicillin G can't get to the peptidoglycan, it can't damage the cell. And so gram-negative cells can survive treatment with penicillin G, and gram-positive cells are killed by penicillin G. Any question about any of that? Well, here's a question for you. Ampicillin is an antibiotic that is one of the early members of the penicillin family that was made. And it kills both gram-positive cells and gram-negative cells. How is ampicillin getting through the outer membrane of the cell wall to get to the peptidoglycan? Anyone know? It's shown in this slide here. All right, can I confirm that somebody's there?
Yes.
All right, great. What happens is ampicillin can get through the porin protein. All molecules that need to get through the outer membrane that the cell wants, that cannot get through the lipid layer or the fatty acid layer of the outer membrane, has to go through the porin protein. And that would be things like glucose and fructose, amino acids. The cell wants those molecules, and they can go through the porin protein, which is sort of like a channel protein. And ampicillin can get through the porin protein, but penicillin G cannot get through the porin protein. All right, let's come back to the slide we were talking about. Come on. Oops, way too small. Around there. So the gram-negative bacteria are resistant to some antimicrobial agents like penicillin G, but it's not true of all antibiotics. Like I stated, ampicillin, which was one of the early members of the family of penicillin to be made, can work on gram-negative cells. And ampicillin also works on gram-positive cells. So we do have antibiotics to work on gram-negative cells. Just state that. All right, let me end up on something that you're not going to be tested on, but I need to explain it to you in case you use this stain for your unknown project. And we'll be doing the unknown project later in the lab. There are special stains, including the capsule stain. And the capsule stain I want to talk about because you need to use it to stain for capsules. You may need to use this stain for your unknown project. So when you apply the capsule stain, it generally will stain both the cell and the background of the slide, but it does not stain the capsule. And this allows you to see the capsule stain. It allows you to see the capsule. The capsule is not visible under normal staining conditions in the microscope. And this right here is a negative stain, staining the cells with India ink. And you see how the background is very dark and the cells are lightly stained. That's why I know this is India ink. And I think this states up here that it is a capsule stain using India ink. The capsules are not stained, but it makes them very visible. All right, any questions about chapter 3? If there's no questions about chapter 3, I'm going to end it here. I will be at the lab at 6:30 to 6:45 to answer questions. If you don't have questions, you do not need to show up.
And then
Yeah.
can you share that PowerPoint again, Dr. Wolf?
Yes.
Thank you.
All right. Bye.